Good afternoon. Today we will be discussing the second topic of bacterial growth and metabolism. Bacteria divide by binary fission. I have told you already in previous class what does binary fission means and does it takes place. The division takes place by the formation of a septum known as a transverse septum. It's nothing but a septum arises in the cytoplasm from the cytoplasm curvings, so which leads to the separation of the two cells, and the septum that develops is known as the transverse septum. The time required for a bacterium to give rise to two daughter cells under optimum conditions is known as the generation time or the population doubling time. The bacterial growth takes place in two ways. Either it increases in the size of the cell as well as the increase in the number of the cells takes place. The total count of bacterial cell is the count of the total number of cells regarding of the cells, either they are living or dead. And the viable count of cells is the count of the cells at a particular moment of only the living cells and that are capable of multiplication. Now, this is the most important part in today's topic. You can see that there is a curve. Yes, it's a bit hazy, but definitely quite readable, as well as the same chart has been given in your books. You can see on the left axis, the y-axis, there is the number of microorganisms, and in the x-axis, it's the time. So after a bacteria is inoculated in a particular media, we need to give time to the bacteria so that it can adapt to that particular environment. Because all the media that we supply, or whenever a bacteria infests in our body, that completely is a foreign environment to the body. So we need to give the bacterial cell a particular amount of time. So during this time, it only feeds upon the particular time of environment and it adapts to the environment. So this is known as the lag phase. During this time, no increase in the number of microorganisms takes place. After a certain period of time, when it has completed its adaptation and has acquired food, it starts to grow. It grows at a huge and exponential rate. This is known as the log phase. This is where the bacterial colonies, colony morphology, biochemical tests, infectivity, etc., all is seen during this period. Because this is the time when the bacteria is growing and it exhibits its pathogenicity too. Now it reaches a stationary phase. During this stationary phase, the growth rate and the death rate almost becomes equal, more or less equal. The viable number of cell count kinds of remain same and tends to go decreasing. So this is the stationary phase. After the stationary phase is over, it reaches to a phase of decline, where the death of the bacterial cell overgrows the growth of the bacterial cell, which leads to the fixed number of total count but a decreased number of viable count. Okay. So moving on to the individual parts, the lag phase. Bacterial cells in this phase increases in size but not in number. I have told you earlier. This phase is just after the inoculation in any media or infection of a particular organism, of particular bacterial cell into a human body. During this period, the bacterial cell adapts to the particular environment. Bacteria starts building up sufficient quantities of necessary enzymes and substances for multiplication. The duration of lag phase varies with species, size of inoculation, composition of culture media, environmental factors, and temperature. Log phase or the exponential phase. This phase follows the lag phase. In this phase, the cell starts dividing. The cell number increases with the time exponentially. This is when colony appears in culture media. The stationary phase. Sorry about the fact that the stationary spelling is a mistake here. Please do correct it. This phase follows the log phase. In this phase, cell division stops due to the nutrients being used for growth and the accumulation of toxic products. The number of daughter cells formed is just enough to replace the number of cells that die. The viable count remains the same or sometimes tends to go decreasing. The phase of decline. This phase follows the stationary phase. In this phase, where the population decreases due to the cell death. There are lack of nutrients and there is accumulation of toxic products. That is why the death takes place. Cell death is also caused by lytic enzyme synthesis. Now moving on to another topic where we will be receiving some classification of bacteria. I will be defining those classification of bacteria, as you have already seen that there are some topics written here. You need to remember these things because as we proceed further, we will be referring the bacteria with these terms on that this bacteria is this bacteria is this. You just need to correlate it. That's why you need to keep these things in mind. Sometimes these individual topics do come as short note in your examination. I mean the university examination as well as internal examination that takes place. So these are the things that we need to keep in mind. The first thing, classification of bacteria based on their nutrition status. These are phototrophs, in which bacteria derive their energy from sunlight. Chemotrophs, where the bacteria derive energy from chemical reaction. Autotrophs, where bacteria can synthesize all their organic compounds. And heterotrophs, in which the bacteria that are unable to synthesize their own metabolites depend on preformed organic compounds. Based on the oxygen and carbon dioxide requirement, aerobic bacteria, which require oxygen for growth, anaerobic bacteria, the bacteria which grow in absence of oxygen. They are two types, obligate anaerobes and facultative anaerobes, grow in absolute absence of oxygen, and facultative anaerobe grows in partial absence of oxygen. Microaerophilic bacteria grows best in the presence of low oxygen concentration, and capnophilic bacteria, which requires an increased level of carbon dioxide, that is 5 to 10%. Based on the temperature required for growth, mesophilic bacteria, cyclophilic and thermophilic. Mesophilic bacteria require around 25 to 40 degree temperature for its growth. The cyclophilic below temperature of 20 degree and thermophilic at a high temperature of 55 to 80. So please keep these things in mind because we will be referring to the bacteria only with these terms later on. So you need to correlate it. Thank you.